Okay, part three, guys. So we're going to talk about proteins, and sometimes you'll see this as peptides, and I don't want you to get confused. So proteins and peptides are formed from amino acids, okay? And when you link together a few amino acids, we call it a peptide. When you link together a lot of amino acids, you get long peptide chain, or we'll call it a protein. And when you get several peptide chains interacting together, you get a protein. Okay, don't get confused. So proteins have carbon, hydrogen, and oxygen. Okay, but you also see nitrogen, and sometimes you sprinkle in a little bit of sulfur. Not always, but sometimes. Okay, carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. All right. So I'm going to go over the basic structure. Guys, this is what happens when you've got a short cord. You're going to get tripped. So this is the basic structure of an amino acid. So NH2, sometimes if you read online, you'll see it as NH3+. We'll go over that later. Don't get confused. That's your amine or amino. You've got this carbon here in the center. It's always attached to an H. The COOH, well, that's your acid part. And then you've got this R group. And R just means a variable. If this was a math class, it would be X. It's another chain that varies. It can be short, it can be long, it can have a lot of OH bonds in there that'll make it polar. It can have a long carbon chain that will make it nonpolar. Okay, this can be a lot of different things. Your body has 20 different amino acids, okay, that we use, but they all vary by this R chain. But they all have these three things. They vary by the fourth, all right? So let me show you two amino acids and how we link them together. And hopefully, if you've seen the first two videos, you're picking up on the pattern. So I just got to shuffle just a little bit. Okay. So there's one amino acid, and it's called leucine. So carbon here, your amine group, H, C, O, O, H, and then you got this carbon chain, 1, 2, 3, 4 carbons, and it's branched a little bit, but it's all carbon and hydrogen bonds. So if we're going over our bonding patterns, a lot of carbon and hydrogen, equal pulling on the electrons in the bond, probably nonpolar. This part of the chain isn't going to interact with water very well. Okay? Over here, same thing, amino group, H, COOH, here's your acid. Well, you've got CH2 and then OH. Oh, it's kind of like an alcohol. This OH portion here, this is capable of hydrogen bonding. It's not just water that can do that. And this is going to be kind of polar. It's going to be able to mix in with water decently. Okay. Well, if we wanted to mix or be able to link these two together, well, maybe you're getting to see a pattern here. This time, though, it matters where you're taking your OH from and the H from, because you can only do it from one place. Look what's close to each other here. Well, you've got an OH here and an H here. Now, again, it doesn't matter which of these two H's you would take. It could be the top or the bottom. Doesn't matter. But you've got to take the OH from the leucine here. You've got to take the H from the serine. Well, if you do it again, that goes bye-bye. Well, now you can link it together. And before I show you that portion, and we're going to black out again, sorry. Hi! So you can get an idea of what these things look like in 3D. Yeah, here we go. Now this one looks a little bit weirder, okay? So this is leucine, and I'm trying to show it so the acid portion is over there, the amine portion is over there. Note that the hydrogen's not pointing directly up and the chain's not going directly down because it's in 3D. Okay. By the way, when we do nucleic acids, I'm not building a model because it's going to take forever. And where do we go over here? Oh, yeah. We can do it this way. So here's your serine. And that blue's not showing up so well. But you got your amine group on the side, your COOH there, here's your hydrogen on the top, and there's that short chain, but that oxygen, your OH there, and so you can kind of see it, okay? So it's still all there. All right, so let's take a look at this thing now. Okay, so you can see where the water comes out. You're missing your H, you're missing your OH, so now the carbon and nitrogen are linked together, okay? This is what's called a peptide bond. And so you're going to see it referenced some in your reading, and you'll see it again during when we're talking about, when we get to units 5 and 6, and we're talking about gene expression and proteins being synthesized and proteins being folded. That's your peptide bond. And sometimes when you see in your reading, you'll see this, the carbon-oxygen double bond and the NH bond circled in there with it. But this is the new bond that gets formed between the carbon and the nitrogen. Everything else is the same. So you could add another amino acid on the backside here, but it would have to bond through an OH of another amino acid. And you certainly could add another one on this side, but you would have to do it through the NH of a new amino acid. Okay? So there's a definite pattern. NH could get added here of a new amino acid, could add the OH over on this side. Okay? You can kind of see the repeating axis. And so here, if you drew it, here's your leucine, and here's your serine. Everything else about the structure is still preserved. Okay? It's still the same thing. Nucleic acids are left.